Thank you, Larry, for inviting me here in this webinar lecture. Today I'm going to share with you the approach that we use in the study of humic chemistry by showing the results of a tomato study and the steps required in order to investigate how the chemical features of humic substances can positively affect the plant physiological response. Now, before we begin to talk, I just want to give you a background about myself. I grew up in Italy, in a suburb outside the city of Naples, where I got my master's degree in agricultural biotechnology from the University of Naples, and from which I further obtained my PhD in management and improvement of natural resources by approaching several studies on both soil humic and organic chemistry, and where I spent 3 years as a research scientist developing new biostimulants capable to boost plant growth and productivity. I made my way up to the United States in 2019 as I started working for biohumanics as soil scientist first and then lab director of humic research. That said, let's jump in our today's talk. When we think about biostimulant, the first product that comes to mind are humic substances, and for a good reason. Indeed, if we look at the biostimulant global market infographics, we can see that humic substances sit on the top tier, being the most adopted active ingredient, therefore having the largest economic impact, followed by vitamins and amino acids, microbials, seaweed extracts, and so on and so forth. And humics are going to hold a major market share by leading a global rising trend, which is expected to hit something like 5.69 billion by 2026, with a major expansion happening in the European countries. However, the fast expansion of the market has led to the introduction of new government regulations requiring the clarification of the mode of action to legitimize the biostimulant industry. As a consequence, the detailed characterization of the chemical composition becomes critical, but at the same time, it gives the companies the opportunity to better understanding their formulations, eventually supplying farmers with effective products whose claims are based on science. At BHN, we formulate and manufacture humic-based biostimulants by studying and screening all available humic sources. Up to 4 top-notch lab spaces under the name Alpha Synectics are available at BHN for product research and development. We can go through the process of extraction, purification, and fractionation up to the detailed analytical characterization. A robust in-house database of the chemical characteristics is built for each source, giving us the ability and the know-how to fine-tune our formulations by implementing only the best humic extracts. The consistency and efficacy of our final products are then tested in several lab-scale and field trials before each new release, and this allows BHN customers to be supplied with premium product selection that meet their specific needs. Now, the structure-activity relationship approach we used consists of several steps. The first one begins with the extraction and purification, followed by the plant experiment, as well as the characterization of the humic material. The chemical features of the extracted humic acids are investigated through several analytical techniques, such as ICP-MS, which reveals the elemental composition, the infrared spectroscopy for the functional group characterization, the carbon-13 NMR, which highlights the carbon distribution, and FT-ICR mass spectrometry to further explore the molecular composition. The information so obtained are used to identify the chemical drivers of humic acids, and along with the data coming from the plant experimental part, the potential relationship between chemical structure and the biostimulant activity is revealed and finally validated by data reduction, correlation, and multivariate statistical analysis. In the specific example I brought you today, we used the randomized complete block design by choosing Microtom tomato for the plant experiment. Microtom is a dwarf cultivar which is perfect for lab-scale experiment as it grows 7 inches tall, but produce little sweet tomatoes, allowing the evaluation of the production at the end of the life cycle. So it's a very convenient model plant to work with. As we wanted to keep the number of variables as low as possible, we decided to use a very neutral substrate such as coconut coir and sand mix in the ratio 2:1. The fertilization used was an Ogland solution adjusted to induce a nutritional stress at lowest dosage. So we went for 100 mpk as fertilizer control and 50 and 25% reduced mpk to induce the stress condition. We applied an ore-extracted and purified humic acid at pre-planting stage at a concentration of 80 milligrams of carbon per liter, so that our final combinations were 25, 50 and 100 as control treatments, and 25, 50 and 100 HA as the humic treated plants. Controlled growing conditions are illustrated on the right. We used a life cycle of 120 days with 14 hours of light and 10 hours of dark. A light intensity was set at 300 micromoles per meter square per second at a temperature of 28 degrees Celsius and relative humidity set at 65%. At the end of the experiment, the production was evaluated by the number and fresh weight tomatoes. Their quality was assessed by measuring the antioxidant content and other parameters such as Brix degree as total soluble solid. Plant fitness was evaluated by measuring heights, the chlorophyll content, and chlorophyll fluorescence as quantum yield, and both root and shoot, fresh and dry weight. Let's focus on the results for tomato yield and quality assessment. In A and B, we have total yield and tomato fresh weight. Both were increased by the application of humic acids, in particular at a low nutritional dose. The yield increased up to 19% and 16% at lower nutritional dose, and at the same time, we obtained an increase up to 24% in tomato fresh weight as compared to control. At full nutrition, the application of humic did not affect the yield, but still increased the fresh weight up to 10%. The total acidity is a parameter often considered as a good indicator for the shelf life, while the total soluble solid is considered a measure of the carbohydrate content. In both, we've got the increase under the humic application. In the case of the Brix, up to 12 and 24% for lower and full nutritional dose. The antioxidant content were also positively affected by the application of humic, in particular under the 50% NPK. To quickly summarize, tomato yield and weight were increased by humic acid treatment up to 19% and 24% respectively. Plant treated with humic acids under stress condition accumulated a higher content of antioxidant. Carbohydrates were also increased as total soluble solids in tomatoes treated with humic acids. In relation to the plant morphological and physiological parameters, shoot weight was increased after humic application at lower nutritional dose, while root fresh weight didn't change so much, possibly indicating the better water retention in the shoots of humic-treated plants. The shoot dry weight confirmed that an increased biomass was developed in humic treatments, and this stress was also reflected in the increased root biomass. A positive trend was shown by the plant heights, chlorophyll, and chlorophyll fluorescence. However, the differences were not statistically significant when compared to non-treated plants. The increase of biomass, despite a similar photosynthetic activity, is an indication of a better nutrient use efficiency in those plants. A better root system and better shoot biomass water retention are indeed indicative of a better nutrient uptake. We used the multivariate analysis to measure the distance between the treated and non-treated plants and investigate the correlation with the measured variables. Closer are the variables to the circled treatments, higher is the correlation. The chart clearly shows a separation of the treatments along the principal component 1 on the x-axis. On the right, we have all the humic treated plant grouped together, and on the left, we have all the controls indicating these two groups are really different. Also, the variables measured fall within the upper right quadrant, where 25HA and 50HA are located, indicating that the responses measured are strictly correlated with these two treatments. Let's summarize the plant fitness outcomes. No differences were observed in plant heights and as well as photosynthetic activity despite a positive trend observed. Humic acid application affected both shoot and root growth. 50HA treatment increased up to 26% the shoot weight, whereas in 25HA we've got an increased root weight up to 32% as compared to relative control. Finally, the multivariate analysis clearly separated the humic treatments from the controls, showing a positive correlation of humic acid treated plants with most of the variables measured, in particular the root dry weight. Let's move to the characterization part, which is the most annoying but at the same time the most informative. The FT-IR is the first step in our characterization process. This is a qualitative analysis, but gives us the fingerprint of the main functional group of our material. This is the spectrum obtained for our material. These two peaks indicate the methyl and methylenic bonds that can be found in fatty acids, waxes, and alkanes. The shoulder below is indicative of the hydroxyl group. This is an important functional group and can be found in alcohols, carboxylic acids, and phenols. In the fingerprinting area, these two peaks are associated to carboxylic acids. In particular, the double bond CO and COH visible here. The high-intensity peak at around 1000 is associated to hydroxyl bound to aromatic rings, such as phenols, and since we are working with humic, most probably coming from lignin material. This peak is associated to CO glycosidic bond that is typical of carbohydrates. In this case, we're talking about cellulose or hemicellulose. Finally, we have the aromatic vibration of CH in the benzene ring, indicating the presence of aromatic material. Each humic acid has a different fingerprint region, and this allows the classification of different material. Here is the carbon-13 NMR spectrum of our material. This is a semi-quantitative type of analysis, but allow us to understand the carbon distribution among different organic molecules. The entire spectrum can be divided in specific regions which are associated to alkyl, methoxyl, carbohydrates, aromatics, phenolics, and carboxylic acids. Then we can integrate these areas below the curve and evaluate how these groups are distributed and calculate a series of indexes that are used to measure the hydrophobicity, the aromaticity, and the degree of lignin as well as alkyl contribution to the stabilization and the humification process of the material analyzed. In the example shown here, we've got that aromatic and alkyl moieties were the most represented in our humic material. The final step of the characterization process is at a molecular level by means of ultra-high resolution mass spectrometry. This is a qualitative and quantitative type of analysis that allow us to identify single molecules and as well assign a molecular formula. This is our spectrum, which shows that more than 13,600 peaks were resolved, of which 9,331 molecular formula were assigned. And on the right, you have a picture of the magnet of this instrument, just to give you an idea of how big it is. Of course, all the molecules identified with this technique need to be organized. So they were grouped based on the hydroatom class and abundance, meaning the presence of oxygen or nitrogen or sulfur, or the combination of nitrogen and sulfur. The abundance followed the order CHO, CHON, CHOS, and CHONS. The average molecular size was 369, and this supports the hypothesis of supramolecular aggregation of small molecules which are associated through hydrogen bonds, pp stacking, and van der Waals interactions. A more informative way to visualize the molecular distribution is to build a Van Crevelen diagram. This diagram cross-plots the hydrogen over carbon ratio as a function of the oxygen over the carbon. This allows us to reveal specific properties such as the oxidation and reduction along the horizontal line, the hydration and dehydration along the vertical line, and finally the condensation and hydration along the diagonal line. These properties are used to define the areas where typical biomolecules can be found, such as lignin, lipids, proteins, carbohydrates, condensed aromatic structures, and unsaturated hydrocarbons. Now, to better understand how these classes are distributed, it's helpful to separate each hydroatom class and plot them individually. CHO is visibly the most abundant class, mainly saturated in the condensed aromatic region, and become dispersed when moving to a protein or carbohydrate region. CHON is laying mainly in the lignin area, with contribution to a small contribution to protein. The sulfur and nitrogen containing molecules are less diverse as the size of the circles increase. Now, the size of molecules in each class can be further simplified by gathering the molecules based on the number of hydroatoms. For example, in CHO, we see that bigger molecules with a higher number of oxygen lie in the condensed aromatic region, whereas the smaller molecules, which are also moderately reduced, fall in the lignin area. CHON molecules are less abundant than CHO, less diversified, but most of them belong to the condensed structures as they are shifted toward the axis intersection. CHOS confirm their low abundance and dispersion along different regions. And finally, the CHONS molecules fall within the condensed aromatic structure with minor contribution to the carbohydrates where smaller molecules are present. Now, the quantification of the molecules lying in each of these regions give us the opportunity to come up with a bar chart like the first that we can see here in A, where we have the general distribution of biomolecules inside this region. As we can see, the most represented compounds fall within lignin and condensed aromatic structures, followed by lipids and unsaturated hydrocarbons. In the chart B, we have the contribution of hydroatom classes to each biomolecule group, indicating, for example, that in the condensed aromatic region, we have mainly CHO and CHON type of molecules. In the lipids, we mainly have CHO type of molecules, and so on with the protein, the unsaturated hydrocarbons, and finally the carbohydrates. In which, in particular, it seems that the contribution are coming mainly from CHOS and CHONS. Now we use this information for the identification of bioactive molecules by means of online databases such as Campbell and PubChem. we matched the molecular formula obtained by FT-ICR with the molecules included into the database and found that the most abundant molecules correspond to flavonoids, quinones, and alkaloid structures. Some compounds, interestingly, are bioactive plant and microbial metabolites. Now, to link the chemical structure to bioactivity, let's talk briefly about plant stress. Plants are constantly facing a stress situation. The mechanisms to cope with stress involve several pathways such as stress perception, signal transduction, and amplification. Now, evidence suggests that humic substances affect the signal transduction by increasing the reactive oxygen species production, as well as the calcium fluxes, as well as the expression of calcium-dependent kinase. Now, to give you a context, let's briefly look at the role of reactive oxygen species. These molecules are produced when molecular oxygen is partially reduced by gaining single electrons and becoming unstable, and for this very reason, reactive. When oxygen gets one electron, a superoxide is produced. The addition of another electron generally leads to the formation of a hydrogen peroxide by bonding to an hydrogen atom. The addition of a third electron splits the molecule into a radical hydroxyl group, which is reactive, and a water molecule, which is not reactive. And finally, a further gain of an electron leads to the formation of a water molecule. Now, these intermediates are called reactive oxygen species, or ROS for short, and they are produced constantly during the aerobic metabolic processes happening in the plants, such as the respiration in mitochondria and photosynthesis in chloroplasts. In particular, in plants, the most active site is the thylakoid membrane, where this process is highly regulated and normally oxygen is released. But when a stress occurs, the photosynthesis could not be as efficient as it normally does, and some ROS are produced. Antioxidant molecules and antioxidative enzymes help converting ROS intermediates to water molecules, making them unreactive. The problem is that ROS can escape these mechanisms and get out of the cell where they alert the neighbor cells that something is not working properly, and therefore triggering further defense system. If ROS are produced in large amount, though, the cells become overwhelmed and unable to compensate the photooxidative burst, and then the presence of ROS starts damaging the DNA, the proteins, and the lipids, by ultimately leading to cell death. Now, ROS are important because they are able to influence multiple vital physiological processing by altering the redox homeostasis. They have a role in the transition from cell proliferation to cell elongation. They play a key role in seed dormancy and germination, as well as they are involved in flower and leaf development. Now, we can think about redox homeostasis as a balance between the ROS production and the ROS scavenging. Under normal growth condition, ROS production is balanced by the ROS scavenging, and we have a normal development. But when the ROS becomes the main event, this equilibrium is broken and the oxidative burst is created, where the presence of ROS and the increase of ROS start damaging the cellular components, such as the membranes, the nucleic acids, and as a consequence, we have a negative stress and a struggling growth. A third option, however, happens when ROS production is higher than ROS scavenging, but does not trigger an oxidative burst. In this scenario, ROS act as a signaling molecules activating the plant's defense system, and we end up having like a positive stress, which is called eustress. Now, our hypothesis is that humic substances are able to modulate redox homeostasis. The modulation can activate the plant's defense system through the ROS signaling, and leading to priming the plant defense system, which respond more effectively, more efficiently to stress condition. Now, the molecules that we found in our study have both antioxidant and pro-oxidant activity. Their role as antioxidant pro-oxidant depend on the biochemical environment and can be transiently antioxidant and pro-oxidant in separate moments. But the idea is that they contribute actively to ROS modulations. So, in conclusion, we can summarize our outcomes as follows. Humic acids in this study enhanced tomato yield and fresh weight under nutrient stress. The treated plants showed a larger contribution, a larger production of antioxidant, indicating that humic acids are able to prime the plant's defense system effectively. Photosynthetic activity did not show differences with the control, but the biomass was increased after the application of humic at low NPK supply, therefore suggesting an increased nutrient efficiency uptake. Humic acid showed a distinct aromatic character where phenolic, carboxylic functional groups were predominant. Condensed aromatic structures and lignin derivatives represented the most abundant molecules. The identification of flavonoids, quinones, and alkaloids within our material is an indication of the presence of antioxidant and pro-oxidant molecules, which support the hypothesis that humic acids are able to affect the plant redox homeostasis by priming a plant defense system through the modulation of ROS production and scavenging. I'd like to acknowledge the National High Magnetic Field Laboratory where part of this work has been done, and giving a huge thanks to Dr. Amy McKenna for the support on and guidance on FT-ICR MS analysis as well as interpretation. Part of these data have been published in Frontier in Plant Sciences, and if you're interested, here is the link where you can read the articles and get more insight into this research. So that's all. Thank you for your attention. This wraps up my presentation. Thanks.
All right, Dr. Monda, we have a couple of questions from the audience, if you'd like. Number one, the process involved in the analysis of humic substances looks quite complex. Why is it so difficult to understand the mode of action of humic chemistry?
Yes, it is. And the reason is because humic and fulvic acids are complex mixtures. This means that the effect that we observe on plant is not the result of a single molecule, but it's rather the result of how different molecules combine together and give rise to new emerging properties that differ from that of the single one, basically. To further complicate the picture, we also have to consider that humic effect takes place in a constantly changing environment, such as the rhizosphere, where microorganisms, fauna, and plant crosstalk can drastically influence the outcome, making it difficult to predict the magnitude of the humic bioactivity.
All right, a second question that we have is, in relation to the hypothesis of ROS modulation, what is the status of your research?
Well, at the moment we are focusing on the condensed aromatic structures of humic substances, and we are investigating their ability to donate and accept electrons, with particular attention to the quinone molecules that trigger a stress event when in contact with plant root surface. The goal is to tweak the humic chemistry in order to control and modulate the plant stress response more efficiently.
All right, that's all the questions that we have. Dr. Monda, thank you very much for your presentation today.
Thank you, Larry, for having me here today. Bye.